Okay, so now that we're recording, before I share my slide, I did want to make one announcement for you guys. Just remember, because this is a summer class, it goes quick. So you guys just took your first exam this past weekend. You should have a pretty good sense of kind of how the class works now, but your next exam is next weekend. And so they come every, 1.5 to 2 weeks for the most part. So I just want to make sure everyone's kind of staying on pace for things and you don't go, okay, well, we took an exam and now we're kind of done, because your next exam comes up pretty quick. Lindsay, you got a question?
Yeah, just a quick question. Are the case studies just like with every exam? So basically they're every other week?
Yes.
Okay.
Yeah, so you pretty much have one case study per exam that covers pretty much most of the material that will be on that exam. So they kind of serve two purposes. One is they help you review the material for the exam. And then the second is they help you learn how to think and work through kind of problems before you have to do that case-based portion of the exam itself. So you don't have a case study running this week. It's not due until next week. You guys are welcome, and this is completely up to you as a group, to meet together and do kind of part of the case study one week and part of it the next, or you could just wait till next week, meet as a group and do it all in one shot, because there is only one per exam.
Okay, awesome. Just wanted to make sure we weren't missing stuff. Thank you.
Chloe, question?
Yeah, I was wondering,
no, you're good.
With our exams, are they like comprehensive? So is like things from the last section going to be on this one, and do they build on each other? And like, is there a comprehensive final? I was just wondering that.
Yeah, so they're not comprehensive. So for the most part, there might be, a question that you can turn on some of the past stuff to understand the wording of the question or things like that. But they're really focused on just the material from, say, this 2 weeks will be on there. For the final, I'm actually doing the final differently this semester, mainly because of the online summer. I wasn't real sure how that was going to work. You don't actually have a final exam time in the summer. So your final exam at the end is actually just going to be a reaction from you guys of kind of know what you learned from the class and how it relates to all the COVID stuff that's going on. So it's more just going to be kind of there's not a right or wrong answer. I just want a sense of what you guys have gotten out of the semester. The case-based kind of exam portions that you were doing is typically, so when I do micro 26 normally, that case-based portion that you're doing for each one of your exams is actually one big exam that I give as the final usually at the end. So it's just a little bit set up differently. So no, the exams won't be comprehensive. You'll just be focusing on what will be chapters 6, 7, 8, and 13 for this coming exam next week.
Okay, thank you.
Other questions?
Just a comment. For me, at least,
okay,
you're
so
cutting a lot.
Looking at chapter 6 and 7.
Can you hear me? Sorry.
Let me. Yeah, I can. Let me check my internet. Make sure that I'm doing okay.
All right. Thank you.
Down here. All right, we'll see if that seems to help at all. Okay, can everybody hear me okay now?
For me, the image, it's still kind of blurry, and I don't know, I have to see you talking a little bit more to be able to tell.
All right, well, I'll share the screen. When the screen is shared, I can't see you guys as easily, so someone just yell out if things aren't working real well on your end.
All right, thank you. Let me...
Okay, so you should all be able to see the screen now. I do have the chat up on the side, so you guys can type in questions there. I'll try to keep an eye on it as we're going if need be. So remember, the key points always start with the learning objectives. This is just to remind you guys what I expect you to be getting out of this chapter and kind of be focusing as you're looking in the book and those types of things. So we're going to start with what is really kind of definition and terminology things. Actually, let me shut off my camera so you're not looking at the top of my head. Okay. Let's see. All right, so let's go ahead and move into looking at some methodology. I always tell people I'm not big on memorize and spit back, but for some of this chapter, understanding the definitions of different classifications, sometimes index cards make a good way to do this, and you just kind of go through and flip through them to get your definition set. It says right, which are all the terminology stuff with description, and you'll notice that when you look at the descriptions themselves, there's only 3 of them, so we're not going to match up all of you to understand the terms themselves. So the first one, I'm going to walk through kind of the logic of how you would do these, and then I'll have you guys answer the next 2 in the chat itself. So this first one says an organism that obtains both their energy and their carbon from glucose. And so first off, we want to say, okay, if we're doing energy classifications, remember to classify based on energy, you can either be a phototroph or you can be a chemotroph. Okay, you're either getting your energy from light or you're getting it from chemicals. Since this says we're obtaining their energy from glucose, okay, that's clearly not light. Okay, so that's going to tell us this one's going to be a chemotroph of some sort. Okay, so as we come through our options, we know it's going to be one of those 4. And then we want to say, okay, what type of chemical is it? Glucose, everybody should know, is organic. So I expect you guys to recognize that things like sugars and proteins and lipids are organic molecules. If it's something weird that I don't necessarily think you would know, I will tell you whether it's organic or inorganic. But those main ones, you should already know that. So since this is an organic chemical, that's going to tell us that this is a chemoorganotroph of some sort. And so we know it's going to either be B or D. And then we need to look at that last piece. So is it an autotroph or a heterotroph? And those classifications are telling us about where it's getting its carbon from. Autotrophs being able to use carbon dioxide to do carbon fixation, and heterotrophs needing to use organic carbon. Since it's getting its carbon from glucose also, and that's an organic carbon source, this one is going to be D. And this is a chemoorganoheterotroph, which is what you guys also are. Sometimes you'll see that abbreviated as just a chemoheterotroph, especially in classes like physiology or general biology, where everything you're talking about is either an animal or a plant. And so there are no lithotrophs in those worlds. And so a lot of times that just gets dropped off. But in micro, we have to keep the organo part in there because there are microbes that don't necessarily use organic chemicals. All right, so we'll look at the next one. It says obtain their energy from photons of light while performing carbon fixation to generate organic molecules. So I'm going to have you guys answer this one. Go ahead, type in the chat whether you think it's A through F based on that description, and then we'll talk about it. Good, seeing some E's show up there, and that's correct. The answer to this one is E. Okay, so they get their energy from photons of light, which makes that easy. This one's going to be a phototroph of some sort, and they do carbon fixation. So remember, carbon fixation was that process of taking carbon dioxide and using it to build organic molecules, things like glucose. Okay. That means that they are a photoautotroph, which was E. And the next one was obtaining their energy from inorganic nitrites while using carbon dioxide to generate organic molecules. So based on that one, A through F, what do you guys think in the chat? Okay, so I see some A's and some F's. So let's talk about this one. This one is A. Okay, so inorganic nitrates for energy makes them a chemolithotroph. And then remember, they're using carbon dioxide to generate organic molecules. Those are those self-feeders. Okay, so those are the autotrophs. So this one would be A. A heterotroph, so a chemolithoheterotroph, F, would have been using organic molecules like glucose for their carbon source instead. Okay, so are there questions on kind of the carbon and energy terminology and how you figure out what might be kind of classified as what?
I have a small question. I think I get most confused with how we classify chemo, just with electrons and stuff like that. Can you just maybe explain that one more time real briefly for me?
Yep. So the key with chemotrophs is so when we say they're getting their energy from chemicals, where that energy is coming from is redox reactions. And so these oxidation reduction reactions that are allowing us to strip electrons from molecules and use those to either power electron transport chains or to generate ATP as we're doing the redox reactions themselves. And so the chemo part just says we're using a molecule of some sort as opposed to a photon of light. The organo or the litho is saying what type of molecule are we stripping those electrons from during those oxidation reactions. And so lithotrophs are using inorganic molecules to strip the electrons. They're the ones that bring electrons to an electron transport chain using something like nitrite or iron, things that you don't typically associate with getting energy from. Hey, you guys don't eat iron to get energy, but bacteria can. The organotrophs are using the organic molecules to do the oxidation. And those are things like glycolysis in the TCA cycle, those types of processes. So it's really about chemo just says we're doing redox reactions using a chemical. The organo versus the litho then says what type of chemical are we using, organic versus inorganic. Does that make sense?
That is perfect. Thank you.
Okay. All right, so we'll move on. Similar concept. So again, matching terminology to kind of descriptions. These are the types of terminology that I didn't go over a lot in the lecture videos. So I talked about oxygen because oxygen's a little bit more complicated, but I do expect you guys to know the classification terminology associated with the other environments. It's just, you don't need to hear me say the definition of this is this. The definition of this is this. You guys can read those. But I wanted to put a couple of them on here so that you get a sense of how these might look as questions and how you could match them up. So I'm going to have you guys answer these. A novel organism that's found growing in natural hot springs that are approximately 60 degrees Celsius. So based on that description, A through G, how would you describe that type of organism? What do you guys think? Good. Seeing a whole bunch of C's show up on there. The answer to this one is C. This is a thermophile. And so when we talk about temperature, you've got a couple different classifications. There's psychrophiles, that are the cold lovers. Mesophiles, which are the kind of body temperature, room temperature lovers. Thermophiles, and then you even have hyperthermophiles that like it even warmer than 60 degrees. When I ask these types of questions, I'll always put a temperature in there. So I'm not just going to assume that you guys know that a hot spring is about 60 degrees Celsius. So you'll always have the temperature to kind of guide you as you're looking at these. The other key, just so you recognize with kind of where the word's coming from, phile means loving. So all these words really mean loving of whatever the first part is. So thermophiles love warm temperatures. All right, let's look at the next one. So an organism growing well at 20% oxygen and then continuing to grow slower when all the oxygen is removed. How might you classify that organism? Okay, so we're going to talk about this one. I'm seeing some B, some F, and some G on there, which are really all of the ones that have to do with oxygen levels. So let's talk about the differences between them. The actual answer to this one is G. Right? This would be a facultative anaerobe. The key with this is 20% oxygen is ambient air. So what you're breathing on a regular basis is 20% oxygen. It's kind of your normal oxygen level. So an organism can grow really well there. What that tells us is that it does use the oxygen in some level. Okay? It's continuing to grow, but at a slower rate when all of the oxygen's removed. So if there's no oxygen there anymore, this can't be F, okay, because F is an obligate aerobe. It has to have oxygen. If you've taken the oxygen away, that obligate aerobe can't grow anymore. So it's like you're an obligate aerobe. If you take away your oxygen, you can live for maybe 5 minutes on the ATP reserves you have, and then you can't make any more energy, so your cells are no longer going to be able to survive. So now we're left with the microaerophiles and the facultative anaerobes. Microaerophiles need low levels of air, so they're an aerophile. They do need oxygen, but they're loving microair, so kind of lower percentages of oxygen. So those actually wouldn't grow well at 20% oxygen. They also wouldn't grow at all if you took the oxygen away. Your microaerophiles, these grow at lower percents, so maybe say 10% oxygen is what they would need. And so this one is your facultative anaerobes. These are the ones that grow great in oxygen, but if you take all that oxygen away, they can still grow. So they've got lots of different metabolic properties that can use oxygen or not. They make more ATP when there's oxygen around, so they do grow faster with oxygen, but without it, they don't die. Okay, so that's the G. And then this last one, a bacterial species that's found in the waters of the Great Salt Lake. And the answer to this one is going to be E. These are a halophile. So halophiles love salt. Okay, so when people talk about the Great Salt Lake and that it's dead and nothing lives in there, they're wrong. There's all sorts of life that lives in there. There's just all microbial life. And so you can go scoop up the water from the Great Salt Lake, and there's plenty of bacterial species that are living in there, but they're all halophiles. They like the salt. If you actually grow them in, say, a normal bacterial media without it, they won't grow. They need that salt concentration there. Okay, so this is kind of how you look at these terms and pair them up with classifications. Do you guys have questions on either any of the terminology on the slide or just any of the kind of terminology classifications at all? Okay, so we'll go ahead and move on, moving into kind of how we quantitate cells then. So here's a question. You've got a patient with a UTI, so a urinary tract infection. The number of bacteria in the urine are counted using both a serial dilution with spread plates and a hemocytometer. So which of the following would be most likely? A, you're going to be able to tell what type of bacteria are causing the infection from the hemocytometer. B, the cell counts calculated for the hemocytometer will be higher than the dilution plating. C, the hemocytometer will allow you to count only living cells. Or D, the cell counts calculated from the dilution plating will be higher than the hemocytometer. So go ahead in the chat. What do you guys think on this one? You might need a second to think about it. But A through D, what do you think the right answer is? Good. Seeing lots of B's show up on the list. And that's the correct answer to this one. This one is B. So the difference between those two, remember hemocytometers, these are direct cell counts. Those are the little fancy slide that has the grid in the middle of it. It allows you to look into the microscope and help actually count them. Whereas serial dilutions, these were doing viable cell counts because they have to be able to grow on the Petri plates. The difference between these is when you do direct counts, oftentimes you're including cells that might be dead. They couldn't grow on the plate, so you don't count them in the viable cell count, but you'll see them under the microscope, so you probably end up counting them. And so that means that these counts are going to be higher because they're including the living plus the dead cells, whereas these are just the living, so it tends to be a lower number. Okay, so that's just understanding the differences between the ways that we can count microbes and kind of what the purpose of these things are. With those viable cell counts, you have to understand how to do serial dilutions and then how to do the calculations for those. So we're going to walk through this problem right now, as opposed to just asking you guys to solve it. I typically say that serial dilutions are the bane of all students' existence. Most students really don't like serial dilutions, so I like to go over them a couple times. All right, so we're going to calculate the number of organisms found in this flask based on this serial dilution and these viable cell counts. And so remember, the equation is the CFUs per mil, which just stands for colony forming units because you're physically counting colonies on a plate. And we want to know how many of those in every milliliter of this flask. And we do that by saying, okay, what were the number of colonies? And we divide that by the dilution of the tube we actually plated, and we multiply that by the amount that we plated, the amount we actually put on the tube. I sent out an email this morning, so in OneNote that accidentally says to add them, and you should be multiplying them. So in the video lecture, they're correct, but in the OneNote, the little equation there has these being added. Make sure you do multiply them. All right, so we're going to fill some numbers in here. First off, we want to know the number of colonies. When we look at these 3 plates that have colonies on them, this one is TNCC. That pretty much just made one big lawn and you can't see individual colonies. But we've got 3 options. Looking at those 3 plates, which one would you pick for your number of colonies? Write the number of colonies in the chat for which ones you would actually use. Okay, so you're going to use the 254. Because remember, whenever we do viable cell counts, we have what I call the magic countable range. And that's going to be, we want between 30 and 300 colonies. That's kind of the statistically accurate range to count in. And so our 254 is between that range. And that's going to tell us that this is the tube here that we're going to want to pay attention to. For the amount plated, notice we've put 0.1 mils on the plate, so we can fill that in. I also want to make sure to recognize this should be in milliliters because we're solving for CFUs per mil. So if this value instead had said 100 microliters, because remember those are equivalent, make sure to convert it to milliliters when you put it into the equation because we are solving CFUs per mil, not CFUs per microliter. And then the last thing we need to figure out is the dilution. So now we need to look at the dilution series. We've taken 1 mil and put it into 9 mils total. And so the dilution of this tube itself is always the amount you transfer in divided by the total amount in the tube. So 1 mil plus 9 mils is 10 mils. So this is 1 to 10. And this series was set up easily. Each one of these is putting 1 mil into 9 mil. So every single one of these tubes is a 1 to 10 individually. But what we know is the original flask. And to do that, you just multiply. So you multiply the dilution of each one of the tubes together, and that will tell you the overall dilution for this 4th tube here was the one we were interested in. So that would be a 1 times 10 times 10 times 10. Okay. When you multiply all those out, this is a 10 to the minus 4 dilution. If we wrote that as a fraction, it'd be 1, 2, 3, 4, 1 to the 10,000th. So that's the dilution that comes into our equation. And then we go ahead and just solve that. And we would get that there were 25,400,000 CFUs per milliliter in this flask. And realistically, what that means is if you pull 1 milliliter out of the flask itself, there are over 25 million cells in that 1 milliliter. That's why if you took that 1 milliliter and put it directly on a Petri plate, all you would see would be one big gigantic lawn because there's no way you're going to count over 25 million individual colonies. That's the whole purpose of serial dilutions, is that our bacterial numbers are so high, we have to dilute them down so that we can actually see them. All right, so questions on calculating CFUs per mil. Anything right now? Yeah, Lindsay, go ahead.
Okay, I just have a clarification on like CFUs. So essentially, we're with a CFU, we're saying that there's one cell that's creating that colony, right?
Right.
So you would say that like at the bare minimum in a CFU, there's one cell, even though there could be like millions in that colony.
Right. So the idea is when you look at a Petri plate, I'll kind of draw it over here on the side.
Yeah.
So, and this is what the reason we say CFUs is because we didn't specifically count cells,
Correct.
but they're pretty much equivalent. So what you're saying with a CFU is that one little individual cell landed on that Petri plate, and then that one little individual cell went through binary fission and made 2, and 2 made 4, and so on and so forth, until you had replicated enough that you've got millions of bacteria in that one colony. But originally, that colony had come from one single cell that had been in your flask. And so we're looking at colonies after growth. Those millions of cells in the colony weren't in the original flask. We assume that if there's one colony there, it's because one bacterial cell from the flask landed on the plate and grew up into that colony. So realistically, each colony that you see came from its own little single individual cell that had grown up into the colony. Does that make sense?
Yeah, so colony forming units is almost the same as saying like a cell. Like if you had 200 colony forming units, you could say there's kind of the same as 200 cells that started the growth.
Yep.
Okay.
Yeah, and that's, we say that realistically, nothing says that there couldn't have been for this colony 2 cells that just happen to be right next to each other. So that's why we don't say cells, because it might be there are a couple colonies there that there were maybe 2 cells that made it, and we don't know that. But realistically, they are equivalent to cells.
Okay, thank you.
Yep. Any other questions?
Wouldn't the dilution number be 10 to the negative 5th because we times 1/10 to the 5th power?
So no, because you've got, so from the original tube, I lost my, so this tube here would be -1. Right, this would then be -2, -3, -4. And so this tube here, we've done, so if you take 10 times 10 is 100, times 10 is 1,000, times another 10 is 10,000. If you actually divide 1 by 10,000, you'll get 10 to the -4.
Right.
Yeah.
Oh, sorry, I accidentally went to the 23 colony. My bad.
Okay. Yep, that's why. Yeah, and that's realistically when I do these, so especially if they're all just 1 to 10s, I really just count the tubes. And so where I just wrote -1, -2, -3, -4, since they're all 1 to 10 dilutions, that's the easiest way to do it. And then you just plug your -4 into the equation. I won't give you guys ones that are tricky and change the numbers on you, so you can kind of get in the habit of looking at that like that. Any other questions? Okay, so we're going to finish chapter 6 by looking at the other math that's associated with the chapter, which is determining how many bacteria would be in a sample after going through binary fission. And so this is the exponential growth equation. Remember, the equation itself is that little b equals big B times 2 to the n. And what all of these things are is little b was number of cells after growth, big B was the number before growth, and then n was the number of generations, which is really just how many times have the cells divided. Okay, so let's take a look at the question. I wrote the question a little bit trickier to make you guys think a little bit about growth curves and how things grow also. Okay, so we've got a bacterial culture. It's in a closed container in the lab, okay, that was inoculated with 100 cells. The cells spend 1 hour in lag phase and then 3 hours in log phase before the population plateaus in stationary. Species has a generation time of 30 minutes, and what we want to know is how many cells are going to be in the population when it reaches that stationary phase. So we're looking to solve for this after growth, so after it's grown and reached stationary phase. But remember, we're looking at bacterial growth curves, so we've got an hour spent in lag phase, 3 hours spent in log phase, and then we've plateaued to stationary. Okay, that's just your standard bacterial growth curve. If we let it keep going, eventually those population numbers would decrease as we hit the death phase. I'm going to start plugging some numbers in, and then we'll work through kind of where we're going here. So we're trying to solve for B. It tells us that the culture was inoculated with 100 cells. So we know that the big B is 100, the number of cells before we were growing. But it doesn't tell us the number of generations. Instead, it tells us the generation time. So we're also going to have to use the equation that generation time equals the time of growth divided by the number of generations. And we can rearrange that because we will need the number of generations. Okay. So we're solving for n here. The generation time we know is 30 minutes. Now, what is the time of growth? And so I actually want you guys to put this one in the chat because this is the reason I wrote the question this way. So we've got the cells spending 1 hour in lag phase and 3 hours in log phase. So to figure out the number of generations, what's the value for t that we're going to use in this equation? Good. So I'm seeing some 3 hours, and I'm also seeing some 180 minutes, which are the same thing. We've just converted. So our time is 3 hours. Okay? And so we wrote this because I wanted everyone to realize that the 1 hour that we spend here in lag phase doesn't get included in this because the exponential growth equation is only happening when the cells are growing exponentially, which is happening in log phase. And so this took 3 hours. So they were growing from here to here. But during lag phase, there's no binary fission. So there is no growth happening. It's why the line is flat. Okay. And so we would use the 3 hours, but our units need to match. So 3 hours times 60 minutes per hour is 180 minutes, which gives us an n of 6. So now we can go back to our original equation. We can plug the 6 in there. And then when we do the math, we're going to get 6,400 cells. And so we would have started with 100 cells here. And by the time we got up to stationary phase, we would have had 6,400 cells. In stationary phase, we still have 6,400 cells. So whether we're talking about here at this time or this time or this time, that number of cells overall in the population has plateaued. But remember, there is still growth happening here. It's just that every cell that replicates, another one dies. So those 6,400 cells are not the same cells. Okay? Some of them have replicated and some of them have died, but now our numbers are going to remain equal. And that's different than lag phase, where we still have 100 cells because they're just not growing yet. They don't start growing until log phase. So questions on either the math, kind of how you do these types of problems, or the growth phases of bacteria? Okay, everyone seems to be good there. We're going to move on to chapter 7. So, oh, Lindsay, you got a question? Go ahead.
Yeah, sorry. So then if the question was posed, like, how long does it take to get to the 6,400 cells, you would add that, the lag phase hour, right?
Right.
Okay.
Yeah. So and there's a question on the worksheet that's like that. If I told you, you started with 100 cells, you now have 6,400 cells, hey, how long were they growing, or how long would you need to actually grow the culture for? You have to add that hour in at the end. So you would pretty much figure out the amount of time they were growing in log phase and then just add your lag phase to it afterwards. Because when you inoculated the culture, you'd still have to wait for that time, even though they're not growing during it.
Okay, awesome.
Any other questions either about this or about anything in that chapter 6 about bacterial growth? Because we're going to move on to chapter 7 now if no one has any questions. Okay, so chapter 7 is the metabolism chapter. Parts of chapter 7 should be familiar to you guys, so you should have a general sense of things like glycolysis and TCA cycle or the citric acid cycle or whatever name you call it, depending on what classes you've taken. And so a lot of the focus is understanding how that previous knowledge now fits into the bacterial world, where bacteria do some weird things sometimes. Okay, so here are your learning objectives. Again, we see kind of the metabolism terminology because we learned the terminology in the other chapter, but now we understand from this chapter why that terminology exists. And so we're looking more at the metabolism behind the words throughout chapter 7. So let's go ahead and we'll start moving through these questions, which are really going to help try to connect those ideas of the terminology and the classifications to the actual metabolism. Okay, so this problem tells us we're working with a chemoorganotroph, and that it's an obligate anaerobe. So again, those classifications that you should now understand what they mean. We've examined the protein in the organism and learned that it does not contain an electron transport chain. So what catabolic metabolism is this organism most likely to use? And we've got a couple options. So go ahead, think about it for a minute if you need to, and then put A through D. What do you guys think this chemoorganotroph that's an obligate anaerobe would be using to get its ATP from? Good. So I'm seeing a bunch of C's show up in the list. Okay. So this one is C. And this is where things get kind of strange. So for your cells can do lactic acid fermentation, but they can only do it for short periods of time. You can't live on fermentation alone. It doesn't make enough ATP. But for some bacteria, they can, and that's all they do for their metabolism. And so when we look and we break this problem down, the first is that we're talking about a chemoorganotroph. That means we're talking about catabolic processes. Remember, the chemo is where do they get their energy from, not how are they building their carbon molecules. What that tells us immediately is this can't be D because carbon fixation has to do with anabolic processes, not catabolic processes. Okay, so that leaves us A through C. We then get to the fact that it's an obligate anaerobe. Obligate anaerobes don't use oxygen, and in fact, oxygen kills them. So they can't be doing aerobic respiration because you need oxygen there, and that would kill them. So that brings us down to B or C, which are both anaerobic processes. So anaerobic respiration doesn't use oxygen, and fermentation can be done in the absence of oxygen. So as we keep reading, we find out that we look at this, and there's no electron transport chain. And that's what indicated to us that this was fermentation. And that's the big difference here, is when you take something like pyruvate, remember, and you're breaking it down, pyruvate can either continue through respiration if there's an ETC, or if there's no ETC, it will go to fermentation. And so the fact that there's no ETC tells us it had to go in this direction, and it couldn't be the anaerobic respiration. And that's the difference between these processes, is both of your respirations here are going to run an electron transport chain. Fermentation does not. Okay, so we're going to stick with our chemoorganotrophs. For chemoorganotrophs, in this instance, because this was an obligate anaerobe that was doing fermentation, this organism would have just gone, say, glucose to pyruvate, and then pyruvate to fermentation. But if they were doing respiration, so either of the other options, there's a lot more steps involved. And so this is just going to be a review. Some of this, depending on who you had for your general bio, might be things you've completely learned in other classes. Some of it might be a little bit new, but I also find students can always use a refresher on respiration. So for this class, I expect you guys to understand what's going in to each one of the stages of respiration and what's coming out. You don't need to know all the individual steps. It's not biochemistry. So we begin with glycolysis. Remember, we get glucose. That gets broken down to 2 pyruvate molecules. And in that process, we're going to have a net yield of 2 ATP and 2 NADH. Remember, this is where that split happened. So those pyruvate can either go to fermentation, where this would be all the energy the cell would make, or it can continue through respiration, where those 2 pyruvate become further oxidized to 2 acetyl-CoA. And in that process, each one of the pyruvate to acetyl-CoA makes an NADH. So we produce 2 NADH molecules. The acetyl-CoA continues on into the citric acid cycle, or the TCA cycle. It's going to combine with 2 molecules of oxaloacetate. And that makes 2 molecules of citric acid, which is where it gets its name, the citric acid cycle, from. Citric acid cycle goes through a bunch of other steps. We cycle back around. This is why it's a cycle. We remake what we started with. And in that process, and we've got 2 acetyl-CoA. So this is happening 2 times for each one of them, and gives us a total of 6 NADH, 2 FADH2, and 2 ATP. And so we've got 2 ATP here and 2 ATP here. At this point, we've made 4 ATP, and this is through what we call substrate level phosphorylation. We've also made a total of 10 NADH and 2 FADH2, both of which would go off to an electron transport chain. Okay, so this is kind of the summary slide of what I expect you guys to know for the what's going in and what's coming out of each of the stages of respiration. Questions about kind of any of the material at this point? Yep, Troy.
I was curious, on our ATP, the NADH, and the FADH2, how much do we need to understand their chemical structure? Or is that for a different class like biochemistry, where that's more important there?
Yeah, I don't need you to know the structures in this class. Sometimes for students, it helps to look at the structures because it makes more sense if you look at the structures and you're like, oh, that's how that change happened, or oh, that's where that electron went. But for this class, as long as you understand what their purpose is, you guys are okay. So in chemistry and in biochemistry, you go into a lot more detail about structures and the enzymes involved in those types of things. For micro, I'm more interested that you understand why we care as opposed to the structural details.
Cool, thank you.
Other questions? Okay. So before we move to that next question, remember those molecules have gone now to the electron transport chain. Okay. That electron transport chain and how that chain functions is key to all of the catabolic reactions we're looking at. So whether you're a chemoorganotroph, whether you're a chemolithotroph, or whether you're a phototroph, if you have an electron transport chain, that electron transport chain functions the same way. Okay, so what's powering the chain, what's the source of electrons and where they're going is different, but the actual chain is going to function the same way no matter what sources we're using. Okay, so I just wanted to make sure that I highlighted that. Okay, so let's look at this question. So now we're studying a chemolithotroph in the lab, okay, and we want to ask kind of the same question we just asked on the next one. Which of the following metabolic pathways does the organism likely rely on for its energy? So go ahead in the chat. What do you guys think? A lot slower on the responses on this one and a lot more mixed. Okay. This is because the chemolithotrophs are the new guys. Okay, so everything we just talked about on the last slide, that's chemoorganotrophs. So you should make a note here that any of these processes, we're talking about a chemoorganotroph. Okay, what that means is that chemolithotrophs don't do any of this. So anything you're seeing on this slide right now, these processes that you learned about in past classes, none of these happen in a chemolithotroph because all of these molecules written here are all organic. So the idea that we got these 2 ATP out of this organic glucose tells us this is a chemoorganotrophic reaction. Our chemolithotrophs don't use organic molecules for ATP. And this is a hard one for students to understand because you're used to thinking about things like carbohydrates and lipids being your energy sources. Chemolithotrophs don't do that. And so the answer to this one is actually A. Okay. Chemolithotrophs are not going to do glycolysis. They are not going to do the TCA cycle. So this can't be all of the above. In order to get their energy, they solely run an electron transport chain. So if we look at that electron transport chain, there's always proteins in the chain that are going to be transporting electrons. And there's always something that's going to drop the electrons off at the transport chain. It's this something that's different between organotrophs and lithotrophs. So for a chemolithotroph, this might be something like nitrate, okay, where it drops them off and forms nitrite. Okay. These electrons are then going to do what they do in every electron transport chain. It's just their source that is different, and it's weird for us to think about because you cannot get energy out of nitrites, okay? But these bacteria can. So that's step one, is drop off the electrons. Remember, those electrons then get passed from protein to protein. And as they do, they're releasing energy. So step two is we're passing the electrons. And the energy they're releasing is used for step three, which is the pumping of protons from inside the cell to outside the cell itself. So this is making our proton gradient, which we call the proton motive force. Okay. Remember, this is outside. Okay, again, there's no mitochondria in bacteria, so this is the actual cell membrane. And then the last step is that the cells need to make sure the electrons go somewhere. They need to come off the chain. And this is also where that something is going to determine whether we're doing aerobic respiration or anaerobic respiration. So if it's going to oxygen, it's aerobic. If it's going to something like, say, iron, then it's anaerobic. Okay. The chain itself functions the same. And so our source of electrons is going to tell us, are we dealing with a lithotroph or are we dealing with an organotroph? If we're using something like, say, NADH. Okay. And our terminal electron acceptor is going to tell us, are we doing with aerobic respiration or anaerobic respiration? Once the proton motive force has formed, that was then used to power ATP synthase to make the ATP. But remember, in bacteria, it's also used for other things. And so we're running these electron transport chains both to make ATP energy, but also to make that proton motive force to use energy to power things like transport of molecules in the flagella. Okay, and so realistically, electron transport chains, their whole purpose is to make this gradient. That gradient then uses its power to do other stuff for the cell. Okay, so that's the difference here between that lithotroph and the organotroph. Are there questions right now kind of about that difference or how electron transport chains work in general? Okay, then the last question is going to focus on what I was just talking about, with an electron transport chain that probably looks different than one you've seen before. So this is an electron transport chain. We've got the inside of the cell here and the outside here. So this is our electron donor dropping the electrons off, and this is our electron acceptor picking them up at the end. And you'll notice here's our hydrogens being pumped to the outside of the cell. Okay, so this is an actual electron transport chain from a microbe. And what we want to know is what type of organism does it belong to? So go ahead, A through D in the chat. Think about it for a minute, and then what do you guys think? Okay, so let's take a look at this one. The answer to this one is actually B. Okay, so we know this is a cell membrane, so it's going to be either B or D. So remember, we're not talking about mitochondria here. This is the electrons are being picked up here by your nitrate and forming nitrite. Okay, that's clearly not oxygen, which is why this is anaerobic respiration. But now we've got that difference between litho and organo. And that was based on the source of the electrons. So where were the electrons coming from? And the electrons were coming from lactate or lactic acid. This is an organic. If you don't know the structure of these, one way that gives that away is notice when it drops the electrons off, what it's forming is pyruvate. And you should recognize pyruvate from this big long list of things where everything on here was an organic molecule. And so when we come back, when we look at this, these are organic molecules. They're not NADH, which you're used to hearing about and thinking about, but they are organic molecules that can drop electrons off. This is actually the opposite of fermentation. So fermentation will take pyruvate and convert it to lactate. This is an electron transport chain that's doing the opposite. It's taking lactate and converting it to pyruvate and dropping off the electrons. But the key is pyruvate's organic, lactate's organic, which means this is a chemoorganotroph. Okay, so it's all about where are the electrons coming from and where are the electrons going. I didn't put a question on here about phototrophs. Realistically, for phototrophs, you just need to understand that it's an electron transport chain. It's running the same as everything else, but the electrons need to be excited before they can actually travel down the chain, and that's what the light is for. So the light's absorbed to power those electrons so that the electrons can then run the electron transport chain, just like every other electron transport chain we've talked about. Okay, so that's it for the slides. So do you guys have questions on anything from the metabolism section for chapter 7? Anything else you want me to go over? Turn my camera back on. Okay. So going once, going twice. Everybody comfortable with the material from both chapter 6 and chapter 7? Last chance for questions about the material or just anything else about the class in general.
I have a general question.
Yeah.
So I was doing some of those online videos of this week 3, and I finished them by the 26th when they were due, and
Yep.
I've gone through to do them again. So I've just hit like, hey, restart it. But when I finish it, it now puts like today's date on it and things like that. So does that, when you look at that, does that tell you now that I've completed on the 28th?
No, you're fine.
Okay.
And so it records the grade book in PlayPosit, only records up till the day that you've done it. So in the grade book, the information will be right, even if it keeps updating on your end where you're looking at it. So you don't have to worry about it.
Some of them say like the 26th, but then I've done another one and that one says the 28th now. And so I was just making sure that you don't think that I guess, didn't complete it by the date or something. Okay.
Yep, nope, you're good. The other thing in the Canvas module on the course materials, I did put a link up recently to the YouTube version of all the videos. So if it's easier where you don't have to deal with the questions popping up and stuff, you can also just watch the straight YouTube video of the lectures and not go to play pause it. If you want to see the questions and review the questions, go to play pause it. But if you just want to watch the lecture, it might be easier to go to YouTube.
Thank you.
Yep. Anyone else?
I also just have a question. The second video that we had to make the comments, for mine now is showing that it's late, probably because I made the comments afterwards. Actually, I just made them a couple days ago. So is that the same case? Would that show that I'm late for that assignment or no?
Oh, no, you're good. So the video discussion comments?
Sorry, yeah, that one.
Yeah, no, you're okay. So the actual assignment will get recorded in the grade book differently than the comments themselves will. So even though you've accessed it and you've done stuff with it, so it updates when you've interacted with it, the grade book will still have it set on my end, so you don't need to worry about that.
All right, thank you.
Yeah. Other questions? Okay. So I will take this recording and put it on Canvas like I've been doing probably sometime this afternoon. And thanks for joining us and participating. And I will see you guys next week sometime.